How do epigenetics explain twin disease differences? Imagine two twins who look alike, grow up in the same house, and share the same DNA. Yet, one might develop a health condition while the other stays healthy. How is that possible? The secret lies in epigenetics. Epigenetics involves chemical changes to DNA and the proteins around it that control how genes are turned on or off. These modifications do not change the DNA sequence itself but influence how genes function. Think of it as a dimmer switch for genes, adjusting their activity levels. When twins are born, their epigenetic patterns are very similar. But as they grow, their environments and lifestyles start to differ. Factors like diet, stress, exposure to toxins, and even infections can cause changes in these chemical marks. These changes are called epigenetic modifications. One common type of modification is DNA methylation, where a small chemical group called a methyl group attaches to the DNA. This can turn off certain genes. Another is histone modification, which affects how tightly DNA is wound around proteins called histones. These changes influence gene activity without altering the underlying DNA code. Over time, these epigenetic marks can drift apart between twins. This process, known as epigenetic drift, accumulates with age and environmental exposure. It can lead to differences in how genes related to immune response, metabolism, and brain development are expressed. In early childhood, the brain is especially sensitive to these epigenetic changes. Experiences like nutrition, caregiving, and stress can modify genes involved in brain growth and neural connections. These changes can shape brain structure and function for years to come. This is why one twin might develop a neurological disorder or other health issues while the other does not, despite having the same genetic blueprint. It shows how environment and experience can influence gene activity through epigenetics. Understanding these mechanisms highlights the importance of early life care. Providing good nutrition, reducing stress, and avoiding harmful exposures can influence epigenetic marks in ways that support healthy brain development. In short, epigenetics explains twin disease differences by showing how chemical modifications to DNA and associated proteins can be influenced by environment and experience. These changes can accumulate over time, affecting gene activity and health outcomes, especially during critical periods like infancy and early childhood.